Enzymes can be investigated by carrying out a practical using amylase and iodine. Amylase is the digestive enzyme that breaks down the carbohydrate starch into the simple sugar maltose. And you can monitor this using iodine solution, which will go from a blue-black color when starch is present to an orange color when it's broken down to maltose. In other words, if the enzyme is successful and breaks down starch, you will observe a color change from blue-black to orange. But if it's unsuccessful and the starch remains, the iodine solution will just remain blue-black. So to set up this experiment, you need to first start off with a spotting tile with drops of iodine in each well. You then set up a water bath using a beaker of water, thermometer and a Bunsen burner to keep the temperature constant throughout the experiment. Next, prepare a set volume of amylase at a specific pH. Then you can mix it with a set volume of starch and start a stopwatch. Now every 30 seconds, use a pipette to add a drop of the mixture to the iodine in the spotting tiles. Then note down the color of the iodine. Repeat this process for several different pHs to see how the pH affects the enzyme activity. If the color of the iodine changes from blue-black to orange, then the amylase has worked and has broken down the starch. You can tell how quickly it's taken to work by seeing how many samples you need before there's a color change. The fewer samples there are before the color change, the sooner the starch has broken down, meaning the better and faster the enzyme has worked. If the solution stays blue-black without changing color, it means the amylase hasn't worked. This is because it's denatured, so the starch has not been broken down. Okay, now let's try a quiz on this topic. See how many questions you can get right. If you liked that video and want to see more, remember to subscribe to Flash Revision Lab to get notified every time we release new content.